Okay, this lecture is about mitochondria and atrial fibrillation. And I'm also going to talk about mitochondrial inhibitors. What motivated me to talk about this was I read a very good paper by this lady named Bianca Brundell. And so usually when I read these papers by, non-native of America, their English stinks, the paper is really awkward in how it's written. But this paper was really well written. I was impressed by it, so I looked her up. So anyways, atrial fibrillation is abnormal, irregular contractions of the atria, which then feed blood into the ventricles of the heart. And if you have abnormal conduction of the electrical impulse through the atria, they don't contract in a coordinated, synchronized way, so they don't fill the ventricles effectively, and you can lose as much as 25% of ventricular filling, which can lead to a decrease in your cardiac output. You'll also get irregular heart rates where you don't have time to allow ventricular filling when it's too rapid. All of this leads to less effective pumping of the blood and can lead to hypoperfusion of the brain and cause dementia just due to that, cerebral hypoperfusion, inadequate blood flow to the brain. Also, the irregular contractions can lead to stasis of blood within this little pocket that sits on the side of the left atrium called the left atrial appendage, and you can form a clot in there which can be tossed out to the brain and cause a big stroke. Atrial fibrillation is one of the most common causes of large major stroke where there's an embolus. An embolus means a clot that moves from the heart up to the brain. Okay, so I'm going to talk about this paper, Mitochondrial Dysfunction and Atrial Fibrillation, because something very interesting came out of this paper. Okay, I'll show you what's going on here. Oh, and first of all, I got to talk to you about something that happens with diabetes, because it's going to be relevant to atrial fibrillation and myocardia in the heart. See, when you're walking, imagine you're walking down a path in the forest, okay? Okay, Peter, Cottontail, hopping down the bunny trail. All of a sudden, coyote. It's like, uh-oh, you got a problem, okay? You need to do two things very quickly. You have to make a decision. What do I do right now? Am I going to fight the coyote? Am I going to turn and run? Am I going to climb the tree? Okay, so your brain just went from, about 10 miles an hour, hopping down the bunny trail, you're all happy, everything's great, no stress, to stressed out to the max, life or death situation. So your neuron has to go from 0 to 100 miles per hour or 5 to 100 miles per hour very quickly. So you have to have a way to ramp up neuronal activity super fast. All right, so that's what I'm going to show you in this slide about diabetes. Simultaneously, your heart has to ramp up its heart rate and its intensity of contractility very rapidly. Okay, so we're going to talk about the mechanism in the brain. In the brain, when you are going to ramp up neuronal activity super fast, the endoplasmic reticulum, which is a reservoir to store calcium, will send calcium into the mitochondria. These are called MAMs, mitochondrial associated membranes. And the endoplasmic reticulum pushes that calcium into the mitochondria, and that calcium, when it hits the mitochondria, it activates Krebs cycle like a spinning wheel, like a gerbil wheel. And Krebs cycle is a pathway that's going to basically take these two carbon units and break them apart and turn them into CO2. And in the energetic process of converting them into CO2, carbon dioxide, you're going to generate electron carriers, something called NADH and FADH2. Those electron carriers will carry electrons to the inner mitochondrial membrane, where they'll be passed along a gradient of electron grabbers to the most powerful electron grabber, which is oxygen. And this whole process is coupled to producing ATP, adenosine triphosphate, the energy currency of a cell. It's like a $20 bill in a cell. Okay, the reason I'm telling you this is that's how it goes in a normal cell. But if the person's a diabetic and they have insulin resistance, they also get insulin resistance in the brain. And the problem they run into is insulin resistance means that when insulin binds the receptor, the normal thing to happen is glucose type 4 transporters are transported from a cytoplasm vesicle up to the plasma membrane of the neuron, and they then open up channels to allow glucose to enter the cell. That glucose entering the cell very rapidly is then converted to pyruvate. Pyruvate then enters the mitochondrial matrix, gets made into acetyl-CoA, and then that's added the 2-carbon unit, is added to the Krebs cycle. So that's how you couple glucose entry to calcium entry into the mitochondria. So glucose entry into the cell is coupled to glycolysis and subsequent pyruvate entry into the mitochondria. And the mitochondria, enzymes are upregulated, made in increased numbers and run in increased numbers to make more ATP. So the point of all this is the blowing a whistle, go, start the race and make more ATP. So these things, all this stuff has to happen very rapidly. So it has to all be in place before the moment, you see the coyote. So here's what happens in diabetes. Because of the insulin resistance, these glucose type 4 transporters don't go up to the plasma cell membrane of the neuron, the brain cell. And meanwhile, the endoplasmic reticulum is not well aware of that, and it keeps on pushing too much calcium into the mitochondrial matrix. When you get excess calcium in the mitochondrial matrix, that can kill the mitochondria because you can start to precipitate proteins, and the cell can then die. If the mitochondria die, then the cell will die. It'll go into apoptosis, depending on how rapid it happens. If it happens super rapid, it'll go into necrosis. That means sudden, abrupt cell death, blood on the carpet, big mess, plasma membrane will lyse, spill into the surrounding area, and it's a big disaster. Okay, it'll more often go into apoptosis, a slower cell death where it recycles itself and the cell is ingested by the macrophages of the brain, the microglia. Okay. But the point I'm saying is because of the insulin resistance, you can't couple glycolysis entry into the cell with calcium entry in the mitochondria. The diabetic person can't ramp up their brain function that well, and these neurons are at risk to die. And this is part of the reason why diabetics tend to be so stupid. Type 2 diabetics are notoriously stupid patients, okay? And that's what it's all about. And we're going to see something similar to this in the heart now in a moment. I wanted you to understand this because I've lectured on this before several times. So when I talk about it in the heart, it'll make sense sooner to you. Okay, so here's the lady who's the leader of this group that wrote this really nice paper. And her name is Bianca Brundel. It's one of these Netherlands or something like that, Amsterdam. Okay, and it was really well written. So somebody in this group writes really well. I couldn't believe how well written this paper was. Okay, mitochondrial dysfunction is associated with a bunch of other things. Oh, there's this guy by the name of Roger Seholt, and he runs a YouTube channel called MedCram. He's a real smart guy. He's out of Los Angeles. He's a pulmonologist. He's also certified in critical care, board certified in critical care, board certified in sleep. So he's pretty good. His nutrition knowledge is okay for a conventional medicine guy. He's getting there. He's improving. But listen to his quote. It was real smart. He said, when mitochondria don't work, nothing works. Yes, that is true. Okay, and I'm going to get back to something why that's so funny. So basically, it's like, your car doesn't work if you don't have any gas in the gas tank. Your cells don't work if they don't have ATP, and mitochondria is where they come from. Okay, look at all these diseases associated with mitochondrial dysfunction. Diabetes, we talked about that. SAT-FAT inhibiting intermitochondrial membrane electron transport. Cancer due to mitochondrial injury, the Warburg effect, let's say from hypoxia, lack of oxygen. Okay, aging, chronic fatigue syndrome. There's an aging theory of mitochondrial degeneration as a major theory of aging. Chronic fatigue syndrome. Dementia and excitotoxicity, Parkinson's disease, congestive heart failure, and many other diseases. Okay, so here is the paper, the abstract, and they say atrial fibrillation is the most prevalent arrhythmia. It's often progressive, can lead to heart failure and stroke and death. Okay, and then here's what's funny. She says the current treatments only attenuate the symptoms, and they are only moderately effective in halting the arrhythmia. So she's basically saying the current treatments aren't very good. Then she says there's an urgent need to understand the mechanisms that drive atrial fibrillation. Atrial fibrillation is characterized by a rapid atrial activation rate, which requires high energy metabolism, and a role for mitochondria is certainly plausible. Okay? All right, so she talks about some things about how cardiac muscle cells function in the atria, atriocardiomyocytes, she refers to them as. And she says that mitochondria take up over 1/3 the volume of a cardiac muscle cell. They got tons of mitochondria, okay? They don't store any significant amount of glycogen, so you have to keep on making more ATP with each heartbeat. And if you're going to accelerate the rate of contractility, for running from a coyote, climbing a tree, then you're going to have to accelerate it a lot, and you just have to pull in more ATP and make it. You can't just break down glycogen because it's not there. Under normal physiologic conditions, the atrial cardiomyocytes in general will mainly burn fatty acids, like 60 to 80%. So let's say on average 70%. Other ATP is going to be coming from burning fatty acids because you're just going along at a regular pace, walking, sitting, it's not a big deal. Your heart's very used to producing that level of energy. When you start running and exercising intensely, you're going to have to use more glucose typically to make that extra ATP for those cardiac cells. You can turn glucose on and off faster than you can fat. Like in the brain, you have to be able to turn things on and off very fast. That's why it just goes with glucose. If you have impaired antioxidant defense due to oxidative stress, all of that will damage the mitochondria. And you're going to be less able to ramp up when you need to, the heart rate and the heart contractility. Let's see, oxidative stress, mitochondrial DNA damage, these happen in atrial fibrillation. So in atrial fibrillation patients, there's already more ongoing reactive oxygen species, oxidative stress. That means when the oxidants outweigh the antioxidants. Okay, and that starts to damage the mitochondria, damages the intermitochondrial membrane, damages the mitochondrial DNA, and leads to these vicious cycle cascades. Okay. Okay, here's a diagram of the intermitochondrial membrane, complexes 1, 2, 3, and 4. Numbers 1, 3, and 4 all pump protons into the intermembranous space. So there's a OMM, outer mitochondrial membrane, IMM for intermitochondrial membrane. The pumping complexes are located along the intermitochondrial membrane. The space in between is called the intramembranous space. Protons are pumped into the intramembranous space. H plus is a proton. Okay, then you build up this gradient of concentrations like a pressurized air, and then when you release the gradient, that yields a lot of energy, and you can use that to make ATP. Okay, so here's the electron carriers coming from Krebs cycle. TCA cycle means the same thing as Krebs cycle, so it's also called the tricarboxylic acid cycle. You feed in the acetyl-CoA 2-carbon units, either taking them off of glucose broken down by glycolysis or fatty acids that are then carried into the mitochondria and undergo a process called beta oxidation, which just means chop them off in 2-carbon units, okay? All right, so anyways, they produce the electron carriers, NADH and FADH2. These send the electrons to be transported down these electron carriers, all the blue protein complexes, and then the yellow H plus is pumped into the intramembranous space. Then you harvest the gradient at ATP synthase, which is also called complex number 5, and you convert ADP, adenosine diphosphate, into adenosine triphosphate, ADP into ATP. And then this is the energy of the cell to do all its work, because you got these high energy phosphate bonds. Okay, and you break apart the last phosphate, and you can get a tremendous amount of work out of that. Okay, let's see. Okay, where it starts getting interesting is they talk about how do you ramp things up. And the way you ramp things up, okay, let me get myself out of here. I'm trying to grab my own picture. To ramp things up, you're going to let more calcium come into this mitochondria. When you got to speed up the heart rate, speed up the intensity of contractility, you're going to take calcium from the sarcoplasmic reticulum. Really the same thing as endoplasmic reticulum. They just call it sarcoplasmic reticulum in a muscle cell. Okay, and these are ryanodine calcium channels is a name for them, but they let out more calcium, and that goes into the mitochondria. Okay, and when this increased calcium comes into the mitochondria, then you ramp up Krebs cycle. You have more of the enzymes available, and they run faster to start running this reaction to make electron carriers and then to produce ATP. So you have to ramp things up again. All right. So that's kind of cool how this is done. These are L-type calcium channels right here that are letting calcium into the cytoplasm. The calcium in the cytoplasm binds to the sarcoplasmic reticulum, and that leads to opening of the ryanodine channels. Then they let out calcium that goes into the mitochondria. That's the major steps in this process. So it's very much like what happens in the brain to ramp up mitochondrial activity to produce more ATP more quickly so the heart can beat faster and stronger. Okay, so I thought that was kind of a cool thing. There's other things involved, heat shock proteins are involved, calcium in the mitochondria. Once inside the mitochondria, Calcium stimulates Krebs cycle activity and ATP synthesis for cardiac contraction. The subsequent increase in Krebs cycle products leads to more NADH and FADH2, the substrates for electron transport chain. Okay. We're also going to talk a little bit about NAD. We're not going to get into NAD in this talk, but it's relevant to this metabolism here. Somewhere else in here it talked about how you have to burn more glucose when you start ramping up cardiac activity. And that's kind of interesting. Okay, so that's what I just wanted to show you. There's an analogous ramping up mechanism, just like you see with ramping up mitochondrial activity in the brain that gets messed up in diabetes. So you have ramping up in the heart, okay? We're going to come back to this later. I'm going to do more lectures on the heart, more lectures on mitochondria, but I just wanted to introduce this topic today because that's a good thing to know. Okay, and I'm going to tell you a few more things about mitochondria. Mitochondrial dysfunction and atrial fibrillation, a little bit about mechanism. Here's another paper. We're not going to go into this right now, but I just want you to be aware of it. There's a lot of papers. Okay, then I'm also going to tell you that I had been aware that mitochondria are a big deal in a lot of diseases, and I knew that, you know, fat, especially SAT fat, was an inhibitor of intermitochondrial membrane electron transport as a main mechanism of insulin resistance with diabetes. And I was kind of curious about what other things were inhibiting the mitochondria. So I have a whole bunch of biochemistry books. I went through all my biochemistry books, and I couldn't find anything on it. None of my college biochemistry books, you know, the old Stryer text, the Leninger text, you know, my other biochemistry textbooks. Nothing about mitochondrial inhibitors. I'm like, something's wrong here. So I said, I got to go to the literature. So I started going to the literature. And by the way, that just shows you they don't teach medical students and college students and biology students, and that's why both your PhDs and your MDs, they don't know mitochondrial inhibition, but it's a super common, important topic. This is typical. Things that are clinically useful, nobody knows them, and they're not in any of the textbooks. Then things that are irrelevant and rare are in all the textbooks, and then to learn clinically useful stuff, you got to go read the medical scientific literature. Okay, so real quick, I found a couple of these guys. Fluoride inhibits complex 4. Hydroxynonenal from lipid peroxidation from omega-6 cooking oils inhibits ATP synthase. Lead, PB, like for plumbism, inhibits ATP synthase. Atrazine, you know, the estrogenic thing sprayed on non-organic corn, it inhibits complex 3. Cadmium inhibits complex 3. Glyphosate, the stuff sprayed on inorganic soy, inhibits complex 2. Mercury inhibits complex 1. Trichloroethylene from the dry cleaner place, liquid paper, that inhibits this right here as well. Some cleaning chemicals, degreasing agents. Okay, paraquat does, rotenone does. All right, so this is just getting warmed up. Okay, we found a bunch of them. And these are common things. Atrazine's all over the place, and most processed food contains atrazine and glyphosate and mercury with high fructose corn syrup. Most people eat oil food every day, hydroxynonenal. Most people drink tap water with F- in it. Most people eat animal foods with sat fat in it. So right away, the average person is automatically eating 1, 2, 3, 4, 5, 6 mitochondrial inhibitors every day. The vast majority of people. That's the minimum. And we're just getting warmed up. Let me show you that stuff I found. I quickly found over 60 mitochondrial inhibitors. Now you tell me, do you think these are common? Tylenol, acetaminophen, okay, metformin for diabetes. These are some of the most common medicines ever prescribed. Statins, inhibiting coenzyme Q. Metformin, inhibiting complex 1. Tylenol, inhibiting complex 3. Okay, tell me if you heard of these other medicines. Lasix, very common diuretic. SSRIs, those are antidepressant medications, inhibiting complex 1. Some of the SSRIs inhibit ATP synthase, okay? And it's not even just them. Some of the tricyclic antidepressants as well, the older antidepressants will inhibit the mitochondria as well. Okay, Haldol, antipsychotic medicine, inhibits complex 1. Chromium can inhibit complex 1. Propofol, the anesthetic, can do it. Antifungals, which are in all the processed foods because they're anti-mold. Antifungal means anti-mold, mold inhibitor. Lots of them are mitochondrial inhibitors. Some real common antibiotics, fluoroquinolones, aminoglycosides, beta-lactam penicillins, mitochondrial inhibitors. Anti-seizure drugs, valproic acid, topiramate, zonisamide, mitochondrial inhibitors that make you stupid, okay? Tetracycline, beta blockers, real common medicines there. Tetracyclines are real common antibiotics. Excess iron, when iron overloaded, that can lead to problems in your mitochondria running the Fenton reaction. Aluminum overload will also be damaging to the mitochondria. Alcohol is damaging to the mitochondria. Arsenic's damaging to the mitochondria. Traumatic brain injury is damaging to the mitochondria. Ingesting silver is damaging to the mitochondria. Okay, this is a lot of stuff. Flame retardants are damaging to the mitochondria. BPA, bisphenol A, the plastic, estrogenic, damaging to the mitochondria. Hypoxia, damaging to the mitochondria. Excess manganese, which you can get from mining or from welding, damaging to the mitochondria. Okay, and there's more. I mean, I'm just kind of getting going here, so take a look at this. A lot of general anesthetics are damaging the mitochondria. That's why I'm glad I've never had surgery. I don't ever want to have surgery. I had a colonoscopy. I did it without anesthetic because I was afraid it would damage my memory. Nicholas Lane, you know, the sort of the brilliant origin of life researcher, he thinks that general anesthetics damaging the mitochondria damage consciousness, and that consciousness arises from the inner mitochondrial membrane. Okay, we're not going to get into that right now today. Phenytoin, another anti-seizure medicine, is a mitochondrial inhibitor. Plavix, the very common anti-clotting drug, mitochondrial inhibitor. Amitriptyline, tricyclic antidepressant. Tramadol, used a lot of times for pain control. Amiodarone, another common medicine, mitochondrial inhibitor. Arachidonic acid, a common fat in animal foods. There's a lot of arachidonic acid in animal foods, omega-6 fat it is, not in plant foods. Okay, so that's not just cooking oil, that's in the meats themselves, like chicken and all that. Okay, so another motivator to only eat single ingredient, whole food, organic foods, all right? To avoid all this stuff. Because, I mean, look how common these are. You can see what I'm saying, that it's very common. People are on 10, even 15 mitochondria inhibitors. Gee, I wonder why they're tired and they don't have a lot of energy and their brain is slow and they're stupid. Okay? And who's the one that figured out all these mitochondria inhibitors are playing a common role in diseases? I did. Okay? I talked to some of the best researchers in the world. I'm friends with them, and they weren't aware of this. I'm the one who figured this all out and wrote about it in my book, Food, Mood, and IQ, as well as my other book to a lesser extent. I had found more of them by the time I wrote this one. So it's good to be simple. Live like Adam and Eve. You know, keep your indoor heating and your indoor plumbing, but eat the things on the earth. You know, like Genesis 1:29. Every plant shall be food for you. That's what God told us. Okay, just a couple more papers. This one's kind of funny. Monosodium glutamate, MSG, is associated with increased risk of atrial fibrillation. Okay, here's just another paper showing MSG is also associated with male infertility, which I think is funny, like as if it's not a coincidence that all this crap that's subsidized is making people infertile and stupid. The aluminum in the water causes leaky gut too, and it can cause a problem with the gonads. Okay, and it's a mitochondrial inhibitor, can increase your risk of AFib. Here is MSG. Omega-3s, all these people are recommending omega-3s. Here's 3 papers showing omega-3s associated with AFib. 1, 2, and here's a 3rd paper. I think that's funny. Here's papers showing atrial fib, antidepressants associated with atrial fib. Okay, tons of people are on all these stupid antidepressants that cause brain damage. Okay, what else? There's a whole bunch of other things. Obstructive sleep apnea is associated with atrial fibrillation. Breathing polluted air every day, atrial fibrillation. Severe infection like pneumonia or sepsis, increased risk of atrial fibrillation. Just being fat, being estrogen overloaded increases your risk. Estrogen overloading can cause amyloid clotting, but I don't got time to go into that tonight. I'll go into that some other time. Mitochondria inhibitors, SERCA inhibitors, all the stuff can cause AFib. So what I'm saying is a typical AFib patient doesn't know what to do. They just take a pill, go for cardiac ablation or something. And what I'm saying is avoid all this stuff. Avoid everything that causes leaky gut. Avoid all these mitochondrial inhibitors. Get your iron down low so you're not iron overloaded. Get your estrogenic chemical load down as low as you can, avoiding all this stuff. There's a lot you can do. Avoid all these stimulants. Avoid alcohol. So the reason to know all this stuff is because you can take action with it and protect yourself to lower your risk of AFib. So let's see if I got any more slides here. Well, that's about it for this talk. Anyways, I hope that was helpful. Give you a couple more ways to reduce your risk of AFib, and if you have it, hopefully stop it from progressing.